Welcome to Analytical Chemistry Vision, a platform dedicated to exploring the theoretical and experimental aspects of chemistry, blending classroom learning with laboratory research to enhance understanding and inspire innovation in chemical science. Let us begin a very interesting and highly useful topic for UG, PG, and research students, second part of the chemical analysis, that is sample treatment. In this lecture, we will learn, after sampling of soil, air, water, food, and clinical samples, next step is pretreatment into measurable form of substance using classical and instrumental methods for their chemical composition determination. Let us see the topics in sample treatment that we will learn here. First one is, need and importance of sample treatment. Second one is, sample dissolution methods for elemental analysis, such as dry ashing, wet digestion and microwave digestion. Third one is, sample dissolution for biomolecules analysis, and fourth one is, fusion process and dissolution of organic sample. Here, we will see how the sample treatment can be defined. Sample treatment is the process of preparing a collected sample for chemical analysis. It includes drying, grinding, digestion, dissolution, filtration, and preservation to ensure the sample becomes suitable, accurate, and interference-free for measurement. What are the importance of pretreatment in chemical analysis? First one is, sample pretreatment removes interfering substances, making the analyte measurable with improved precision and accuracy. And second is, pretreatment stabilizes analytes, reduces matrix effects, and ensures reliable results in all analytical applications prior to instrumental measurements. The sample treatment of soil, water, air, and food samples helps convert metals such as copper, iron, zinc, sodium, potassium, calcium, and magnesium, as well as anions like sulfate, nitrate, phosphate, and chloride, into their free forms in solution for accurate measurement using analytical instruments. Similarly, fruits, grains, cereals can be also treated to determine the biomolecules such vitamins, proteins, fats, flavonoids, phenolics. Important step in sample treatment is sample dissolution method for elemental analysis. Sample dissolution for elemental analysis involves converting solid samples into clear, homogeneous solutions suitable for instrumental measurement. Proper reagent selection, controlled temperature, and contamination-free procedures ensure accurate and reliable elemental quantification across diverse sample types. Methods include acid digestion, fusion, and microwave-assisted digestion, which break down matrices and release target elements. First one is dry ashing. Second one is wet digestion, third one is microwave digestion, and fourth one is fusion. Let us see first method for sample dissolution for elemental analysis, that is dry ashing. Is a sample preparation technique in which organic materials are decomposed by heating them at high temperatures, 450 to 600 centigrade in a muffle furnace. The organic matter burns off, leaving behind inorganic ash containing the analyte. This residue is then dissolved in suitable acids for elemental analysis. The method is simple but requires careful control to avoid volatilization losses of sensitive elements. Here, we will see the methodology for dry ashing of soil, vegetables and food samples. First one is, weigh 2 grams of the powdered soil, food sample and dry it at 105 degrees Celsius to remove moisture completely. Second one is, transfer the dried sample into a clean crucible, gently pre-ash to remove organic matter, then ash fully in a muffle furnace at 500 centigrade for 4 hours. Third one is, cool the crucible in a desiccator and weigh the remaining ash. Fourth one is, add 10 milliliters of concentrated HNO3 to the ash, heat the mixture to fully dissolve the minerals, and then dilute to 100 milliliter with distilled water. Finally, fifth one is, the filtrate obtained is used for metal analysis using AAS, flame photometer, ICPOES or ICP-MS. Here, what will be the advantages and limitations of dry ashing method? Advantages. Simple, inexpensive equipment, muffle furnace. Good for many metals in matrices with high organic content. Produces ash suitable for acid dissolution and instrumental analysis. Limitations, time-consuming, hours. Not suitable for volatile elements. Potential losses or contamination if poorly controlled. Some soil minerals resist dissolution, fusion or HF digestion may be needed. Next, here we will learn what is wet digestion method. This is a chemical method used to break down complex organic matrices such as soil, food, milk, and vegetables using strong oxidizing acids. Concentrated acids like nitric acid, perchloric acid, sulfuric acid, or hydrogen peroxide are heated with the sample to dissolve organic matter and release elements into solution. This technique provides complete digestion, minimizes elemental losses, and produces a clear solution suitable for elemental analysis by AAS, ICPOES, or ICPMS. It is widely used for accurate determination of nutrients, heavy metals, and contaminants. Next, the methodology for wet digestion of soil, vegetables, and food samples. First one is, weigh 0.5 to 1 gram of fine powdered dried vegetable, soil or food sample into a digestion flask. Second one is, add 10 to 15 milliliter of concentrated acids such as nitric acid alone or mixtures like nitric acid and perchloric acid or nitric acid and sulfuric acid. Third one is, heat gradually on a hot plate until vigorous reaction subsides and fumes clear. Fourth one is, continue adding small acid portions until the solution becomes clear and colorless. Fifth one is, cool the digest, filter if necessary, and transfer to a 50 or 100 milliliters volumetric flask. Fifth one is, dilute to mark and determine metals using AAS or ICPOES. Here, we will know what will be advantages of wet digestion method, which are given below. Efficient dissolution of organic matter in food and soil samples. Suitable for a wide range of metals, iron, zinc, copper, lead, cadmium, nickel, etc. Requires simple, inexpensive laboratory glassware. Produces clear solutions ideal for AAS, ICPOES analysis. And then limitations of this method are: time consuming compared to microwave digestion. Risk of contamination or metal loss due to open vessel heating. Strong acids, nitric acid, perchloric acid, sulfuric acid, pose safety hazards. Requires careful temperature control to avoid explosive reactions. Third one is, microwave digestion method, which is an advanced sample preparation technique that uses controlled microwave energy to rapidly digest soil, food, milk, and vegetable samples in sealed, high-pressure vessels. A mixture of strong acids, typically HNO3, H2O2, or hydrofluoric acid, HF, for silicate soils, is added, and microwaves heat the solution uniformly, achieving higher temperatures and pressures than conventional wet digestion. This ensures complete decomposition of organic matter, faster digestion, 10 to 40 minutes, minimal contamination, reduced reagent use, and improved recovery of trace elements. The resulting clear digest is ideal for precise elemental analysis by AAS, ICPOES, and ICPMS. Here we learn the methodology for microwave digestion of soil, vegetables and food samples. Firstly, weigh 0.5 to 5 gram of dried fine powder depending on concentration of metals, vegetable, soil, food sample into a Teflon digestion vessel. Second is, add 6 to 8 milliliters HNO3 and 1 to 2 milliliters H2O2 to oxidize organic matter efficiently. Third one is, seal vessels, place in the microwave system, and run a programmed cycle reaching 180 to 200 centigrade for 20 to 30 minutes. Fourth one is, allow vessels to cool completely before safely opening. Fifth one is, transfer the digest to a flask, rinse the vessel, filter if needed, and dilute to 25 to 50 milliliter with distilled water. Analyze metals using AAS, ICPOES, or ICPMS. Let us see the advantages of microwave digestion are: rapid digestion with significantly shorter processing time. Higher temperature and pressure ensure complete breakdown of soil, food matrices. Closed vessels minimize contamination and loss of volatile elements. Requires smaller volumes of acids, making the method safer and cleaner. Produces clear digest suitable for AAS, ICPOES, and ICPMS analysis. Limitations of this digestion method is: requires expensive microwave digestion equipment and Teflon vessels. Finally, fourth method is fusion process for dissolution of organic samples, where fusion is a high-temperature decomposition process used to break down refractory or organic-rich samples by heating them with fluxes such as sodium carbonate, potassium hydroxide, or lithium metaborate. This method is used because the dry ashing, wet digestion, microwave digestion methods unable to break down the refractory or organic-rich samples. Here we will learn about the methodology for fusion process. First one is weigh 0.5 to 5 gram of the fine powder dried organic sample into a platinum, nickel crucible. Second one is, add a suitable flux such as Na2CO3, KOH, or LiBO2, in excess. Third one is, heat slowly to remove moisture and char organic matter. Fourth one is, raise temperature to 600 to 900 centigrade until a clear molten fusion is formed. Fifth one is, cool and dissolve the solid melt in dilute acid or water. Sixth one is, transfer and dilute the solution for elemental analysis, AAS or ICPOES. Next one is, enzymatic digestion which used for biomolecules analysis. This method is a sample preparation technique in which specific enzymes are used to break down complex biological, vegetables, fruits or food samples into simpler, soluble components. The enzymes, such as proteases, amylases, or lipases, selectively degrade proteins, carbohydrates, or fats without harsh chemicals or high temperatures. This gentle process releases target biomolecules like vitamins, proteins, carbohydrates, minerals, or bioactive compounds, flavonoids, caffeine, and phenolics, etc. into solution and analyzed using HPLC, LCMs. Here we will understand the application of enzymatic digestion for biomolecules determination. First one is vitamins determination, particularly B complex, B1, B3, vitamin C, and fat-soluble vitamins after TCA diastase treatment. Next, carbohydrates or sugars, glucose, maltose, lactose after amylase, invertase, or lactase digestion. Further, proteins or amino acids, total protein, peptides, and amino acids after protease digestion. In addition, lipids or fatty acids, released and quantified after lipase digestion. Further application of enzymatic digestions are phytochemicals, polyphenols, flavonoids, carotenoids made more extractable after enzyme treatment. Minerals such as calcium, iron, zinc, released from protein mineral complexes by enzymatic hydrolysis. Dietary fibers, soluble and insoluble fractions measured after cellulase, pectinase digestion. Nucleic acids, DNA RNA fragments in molecular biology after nuclease digestion. Here, we will learn about the methodology for enzymatic digestion of vitamins from food or vegetable samples. First, take 1 to 5 gram G dried food fine powder sample into the flask. Second one is, add 10 to 15 milliliter acetate buffer, pH 5.0, prepare slurry. Third one is, prepare TCA diastase enzyme solution, 2% in the same buffer. Fourth one is, add enzyme to slurry and incubate at 45 centigrade for 60 minutes. Fifth one is, heat mixture to 80 centigrade or add ethanol to stop enzyme activity. Sixth one is, centrifuge and filter the supernatant to obtain clear extract. Seventh one is, dilute appropriately and analyze vitamins using HPLC, UV visible spectrophotometry, or LCMS. These are the given references for making this video lecture. Thus, in summary, this lecture outlines essential steps of chemical analysis, where the sample treatment resulted the measurable form of chemical substances prior to instrumental analysis. It is highly useful for students doing dissertation or research work, as well as chemists working in industries and testing laboratories. The content strengthens understanding of accurate samples preparation and analysis of environmental, food, and clinical samples. In next, third part, steps in chemical analysis, we will learn the use of analytical instruments such as atomic absorption spectrometry, AAS, UV visible, inductively coupled plasma optical emission spectroscopy, ICPOES, ICPMS, HPLC, and LCMS for measurement form of chemical substances, like metal ions, anions. Pesticide, biomolecules, vitamins, fats, proteins, volatile organic compounds, benzene, toluene, with the help of sample treatment. Thank you for watching the Analytical Chemistry Vision. If you like this video, please visit our channel for more interesting theoretical and experimental lectures. Don't forget to like, share, and subscribe, and share your feedback in the comments to help us improve further. Thank you.